Yeah.
Hi again, we're here. Questions about the way the course is going? Thank you, someone, for pointing out to me that D2L claimed, of course, now I would get a phone call, that D2L claimed we were going to have a test this coming weekend, but I've been saying it's not. I'm right. D2L was wrong. It is my goof. I had put the wrong date for when the exam will be available to you, and D2L puts it in the calendar automatically. Okay, so the exam is not this weekend, it's the next weekend.
Yeah, I thought it was last Thursday, so I took another picture.
I, as it happens, I did today. Yeah. Other question. The question was, had I uploaded last Thursday lecture recording, and I did. And so I've been quietly putting all the lectures on D2L, recordings of them. I don't want to advertise that a lot because I want you to come to class. And indeed, you'll be in cahoot trouble if you don't come to class regularly. Anyway, so far, seems like attendance has been pretty good, especially with, I got at least 5 people today telling me they have COVID. So if you aren't being careful, think about that. Other great questions. Okay, so we've been talking about the metazoan's dilemma. How do you make a body that's got lots of different kinds of cells in lots of different places and the place such that they can coordinate together and do what natural selection wants, which is make babies? And we've seen that in fruit flies, they start off the embryo is a syncytium, where all these individual nuclei, no bothering with cellular divisions between them. And first and foremost, there are maternal polarity genes where mom, before the egg was fertilized, had dictated which part was going to be the anterior end and which part was going to make the posterior end by the glomming of various factors like nanos and bicoid and things like that. And we saw that these are transcription factors. And we saw that once you have just 2 different factors, like these maternal factors, bicoid and caudal, that it's not unusual for these transcription factors to have opposing effects on gene expression, so that cells that are being stimulated by one are being inhibited by the other. And so that means that you can end up with with just those 2 factors, you end up with a continuum that defines the anterior-posterior end of the animal, or the animal it's going to be. So already the nuclei have been told where they're going to appear in the body plan. And eventually cellularization happens, and by the time that does, right, each one has already gotten their marching orders. So once you have transcription factors that are interacting with each other and opposing each other, acting in antagonistic fashion, then they can do things like there will be another layer of transcription factors that are affected by those. And then you end up with strange things like this, where there's quite a lot of this hunchback up to this point, and then it drops off very suddenly. So the work of these 2 different transcription factors is causing more of a demarcation and more of a limitation of another layer of transcription factor. And this is one of the so-called gap genes. And the gap genes in turn are going to affect the later expression of a different set of transcription factors, the pair rule genes, which are then going to eventually bring about the transcription of yet another set of factors, the segment polarity gene. And in this way, that syncytium gets divided up into smaller and smaller pieces, each little region of which has a unique developmental history of what transcription factors it's made, and therefore what gene expression it has been putting out. So already each has a little set of orders. You're going to be in the head. You're going to be in the abdomen. You're going to be in the leg. Everyone see that so far? Pretty clever. Natural selection. And I just want to point out to you that the gap genes and pair rule genes are indeed transcription factors, but by the time we get to the segment polarity genes, now cellularization has taken place. And so the segment polarity genes are not themselves transcription factors. They are not the ones that are going to bind to DNA. Rather, they're signaling proteins between cell membranes, and they influence gene expression in each other. So one cell is expressing one of these, segment polarity genes, and it's communicating to a nearby cell, and that communication changes gene expression in that cell. So far, so good. And these are hierarchical, I said, in every sense. There's fewer maternal polarity genes than any others. So as you go down more and more, you need more members of each class in order to divvy up the body more. And they also appear at different points in development. So the maternal polarity genes are there even before it's fertilized. And they're going to influence, et cetera, et cetera. So, and here again, I'm pointing out that the segment polarity genes use membrane-bound signaling. There is, of course, yet another set of genes that are indeed transcription factors that are going to be regulated by this cascade that happened to them. And they're known as homeotic genes. We'll talk about why they're called that in a moment. And here's a more graphic representation. So here, these are different embryos killed at different times and looking for members of different parts of the hierarchy. So notice the maternal polarity genes are being expressed at the very beginning and the others aren't. But as development proceeds, now the maternal polarity genes are being expressed, but so are the gap genes, but the pair rules aren't. We go a little later in development, gap genes are being expressed, and now pair rule genes are being expressed, but not the segment polarity genes. And so we go on like that until at the latest in development, we have this situation where the segment polarity genes are being expressed too. And so again, every nucleus, every cell has a relatively unique history of transcription factors and gene expression. And I say relatively unique because, of course, two cells that are right next to each other are, of course, because of where they happen to be, they're going to have a very similar history. Questions about that? Look at that. We just made a body. We gave instruction. We figured out how to be a metazoan, where you have lots of different cells in lots of different places, and they're all going to take on a different fate and make the thing you need to make babies. Simple, right? Here's a table to go over those various layers that we're talking about. In addition to the homeotic selector genes, typically known as homeotic genes, or as we'll see in a moment, Hox genes, there are realizator genes. In fact, there's no standard term for this. I just want to point out to you that once we get to this stage where these transcription factors are at work, that in order to actually tell the cell, make a liver, make a leg, make an antenna, in order to tell the cell that, it has to be regulating another set of genes that actually make the structure that you're going to end up with. We're just talking about the genes that are in control of that organization, not the genes that actually make the structure. That's yet another hierarchy of genes, and there are lots of those, as there would have to be, because they make lots of different structure. To explain to you what homeotic, and here's a mnemonic you can use or not. You can make up your own, whatever it is. So maternal polarity, gap genes, right, etc. Here's an interesting thing. We know about all these genes, but in this hierarchy, but the first genes we learned about were way down here, the homeotic selector genes, because it turns out, as we're going to see in a moment, mutations in these genes in flies sometimes result in truly bizarre phenotypes that are obvious. And the reason they got our attention is because, when they were doing those screens, purposely mutating lots of flies, etc., they were getting mutations in these other genes but almost all those mutations are fatal. And so no larva ever was produced. And yet it's going to be these genes that act latest in development, where mutations there are not quite fatal. They just result in something strange. And so a larva is formed, an adult comes out, and the experimenter is sitting there blinking at the fact that this fly's got a leg sticking out of its head. Okay? I point this out to say, here's an example of how mutations early in development are very likely to be fatal, right? Whereas mutations of genes that are happening later in development have at least a chance of sticking around. Why? Because you've been messing with the bread dough, right? Or the donut dough. If you don't put in the yeast, you're in trouble. How's that? Okay, let's talk about some of these. Oh, no, I have to ask for questions because before I show you these bizarre mutations, these homeotic mutations, any questions? Let's go. Okay. These are the things we're going to learn. And now we're going to talk about homeotic genes. Of all those genes I've told you about, they were the ones that were first discovered, some of them in the 40s and 50s, through just blindly mutating flies and seeing what you get. And once you did get a cool mutation like that, they knew how to map the chromosome and figure out where it was. And as it turns out, there are lots of these different kind of weird things, and they were all on the same chromosome. And here's one, the so-called antennapedia, where the fly, instead of making an antenna out here, it's making a foot. You know, antennapedia, like foot. And so these, once they started cataloging these, mostly by a guy named Lewis at Caltech, once they started cataloging these, they called them homeotic selector genes. And they called them that because it was homo, like an entire thing, and eotic as in structure. So it's like a whole structure got swapped. In this case, an antenna got swapped for a leg. Or situations like this. If you look at fruit flies closely, you see they only have two wings, and that's because in their thorax, the first of the three segments of the thorax makes wings, and the next two do other things. Well, bother with what? But in some of these mutations, they had two first segments of the thorax. So what should have been the second segment of the thorax duplicated what was in the first set of the thorax, and they ended up with two sets of wings, so-called bithorax mutation. I'm probably covering that up. In this case, ultrabithorax. And here's another view of the same sorts of flies. And as I said, they cataloged a lot of these homeotic genes. And eventually, by the 1970s, they were able to clone and sequence these genes. And they found not only were they all in the same chromosome, they all had a part of the gene that was the same, a part that they called the Homeobox domain. It was a stretch of genes that made the part of the protein that liked to bind DNA. And so this was one of the early indications about transcription factors. So one of the first transcription factors that were sequenced were these Hox genes. And let's see. And then there's a bit of terminology that I want to try to warn you about. So the Hox genes are indeed transcription factors. And when they found the Homeobox domain in these Hox genes that were all in the same chromosome, they saw, holy cow, that same domain occurs in lots of different genes, not just on one chromosome, but all through the animal. And they reasoned that that meant that these genes were also making transcription factors. And so they started calling them Homeobox genes. So we have this weird situation that there's genes that are called Hox genes, and they all have a homeobox domain because they're all transcription factors. And what makes them a gene is that they are in this place on the particular chromosome, I'll show you where in a moment, and that they tend to swap out body parts. But then there were other wannabe genes that also had the homeobox domain, but weren't Hox genes. Mutations there never swapped out body parts. Everyone get that? And they're all transcription factors, but I want to warn you, we're going to run into lots of transcription factors, right? And so the set is, there are loads of transcription factors in the genome, genes that make proteins that regulate gene expression. Some of those transcription factors are homeobox genes, and some of the homeobox genes are Hox genes that have those dramatic effects. So I was paying attention. I was studying mammals. I wasn't studying flies, but I'm paying attention to what the fly folks are doing, going to seminars and stuff like that. And I can tell you that once they found the homeobox domain, there was this weird thing that they noticed, which is all the homeobox genes were on the same chromosome, as I said before. I actually forget, maybe it's chromosome 4. Anyway, they were all on the same chromosome, and where they fall on the chromosome correlates with which part of the embryo they get expressed in. And so this was called collinearity of Hox genes. And to this day, no one knows why that happened. But as we'll see in a moment, it can't be a coincidence, right? There's something important about their placement on the chromosome for their work. And here are the various names of them. So far, so good. And the other thing, as a mammal guy, while the fruit fly people, and there's always, some sort of competition between scientists, right? And so always the folks studying mammals say, well, those guys are studying flies, but what's that got to do with humans? I'm going to study mammals because they're human. And then they study, mice. Well, you could ask the same, well, what have mice got to do with people, right? And other people say, well, mice, what do they have to do with people? I'm going to study monkeys because they're the closest to, you see the idea. And it's all stupid, right? So at the time they're making these remarkable discoveries about mapping out the body of fruit flies, everyone in the mammalian world, not everyone, many people were dismissing it. It's got nothing to do with mammals because mammals aren't segmented. You look at a fly and you can see the segment, right? It's not quite as dramatic as when you look at an earthworm, but you can see it's made up of segments. People aren't made of segments. And as a guy that was studying spinal cords at the time, for me, it was like, what are you talking about? Of course we're segmented, right? We even call them spinal segments. See, the nervous system is segmented in humans, right? And what's happening is some parts of the nervous system are a whole bunch of segments that got squished together to make a big thing called a brain. And we already knew that in flies, what was going to make the brain starts off with individual segments that grow a whole bunch and get smushed together. Smush being the technical term. So but we're so used to seeing people the way they are. I mean, you can tell the segmentation is more obvious when people are like that, like our four-legged ancestors were. So guess who was pleased? I had nothing to do with it. I didn't do it myself. Guess who was pleased when they looked for Hox genes in mammals, and lo and behold, they were there. And just as, one big difference between Hox genes and flies and mammals is that in flies, they're all in a single chromosome. In mammals, they occur on 4 different chromosomes. But in mammals, as in flies, and it's most obvious in the spinal cord, the sequence in which the genes occur on the chromosome is matched by the sequence in which they are expressed in the developing embryo. In other words, there's collinearity there too. So all those people who are dismissing what the fly people are doing, they were proved, really wrong. And not one of them apologized. They said, oh, well, okay. Now we'll study Hox genes in people. The fact that there are on 4 chromosomes in humans means there's a terminology problem. And so people started talking about the HOX-A genes on chromosome 7, HOX-B, HOX-C, and HOX-D. You can tell the order in which they were discovered, which was not the same as the order of the number we give to chromosomes. And this is an important part of evolutionary biology that you've run into before, but let's talk about it in terms of making a body plan, and we'll run into it several times in making a nervous system, which is what sometimes happens in evolution is that a particular gene will get duplicated. And the presence of 4 different chromosomes with Hox genes suggests that there are sometimes when an entire chromosome gets duplicated. Most of the time when such mutations happen, the individual is dead and no one ever hears from him again. But sometimes there'll be a duplication of a gene that is not fatal. And for a while, the organism and its offspring and its population will have 2 copies of the same gene and they're doing the same job. But an opportunity arises when you have 2 copies of a gene that's crucial, which is a mutation in one might not kill you. And so what happens over time is mutations accumulate in one of the copies, doesn't have to be the original, doesn't have to be the arrays, and they start fulfilling different functions. Natural selection ends up using them to take on a different, and over time, the 2 genes come to be quite different from each other structurally and may serve totally different functions. Everyone get that? And so that seems to be what happens in humans, that there are these 4 sets of Hox genes. Not all of them are found in every chromosome, but enough of them are, and they're expressed collinearly. This probably reflects the difference in complexity to make a human being versus a fly. Once you know more about fly development, you see that they are really, truly trimmed down, lean, little yeast-eating machines, right? They're really good at that. And they're not that complicated, right? Not that complicated nervous system. By the time you're going to make a mammal, you've got to make a lot more brain parts. You've got a lot more sensory systems to deal with. And you've got a lot more complex decisions, blah, 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 a lot more complicated motor programs to work out, et cetera. And so probably the additional, the duplication of Hox genes in mammals probably reflects what's required to make a more complex body. Because think about it, more complex body means you got to specify more different parts in a human than in a fly. So far, so good? You're just teasing me. You're going to act like you're about to ask. Anyone? Okay. Most mutations in Hox genes, let's see. There are very few, no, I don't even start that way. There are mutations in Hox genes in mammals, right? They do happen, but because of the Hox genes being on 4 different chromosomes, a mutation in one of the Hox genes isn't always fatal and may not even show much of a phenotype. And so here's an example where we're looking at 2 different Hox genes that are important for developing a limb in mice. And here's what wild-type animals look like. If you knock out one of the Hox genes, HOX-A, you can see there's definitely a deformation of the limb there, but it's mostly okay. And if you have a wild-type version of that Hox genes, but then knock out the other, then it looks even more normal. But you knock them both out, and you don't end up with a limb, right? So there's some redundancy here in the system, which may also be why there are 4 sets of Hox genes in humans and mammals. Then, yeah. Question? So just to flog the horse of how segmented we are, I'll just remind you that during development, we hardly ever talk about the spinal cord, but looking at the brain, pretty early in development, the neural tube grows differentially, dividing faster in some parts than in others, et cetera, and moving around. And so we have these segments, and we do our neuroanatomy. We talk about these segments, right? There's early in development, we say there's a forebrain, which is eventually going to give rise to the telencephalon, cortex, and the diencephalon, hypothalamus and thalamus. And then there's the midbrain, which doesn't grow that much in development, and so it makes the relatively tiny midbrain. And then there's the hindbrain, which will later get divided into the metencephalon and myelencephalon, pons cerebellum, and the medulla. Pretty segmented. And in mammalian nervous systems too, it's the various Homeobox genes, Hox genes, that are going to lay out the rostral-caudal function of the nervous system, where they're going to be working to tell each cell, this is the part of the nervous system you're in, and therefore that's the developmental pathway you need to take. All this stuff presumably is reviewed for you. So here's some examples of Homeobox gene expression in the nervous system, and we've just graphed it out, and I've not specified when during development it happens because there's enough detail already. So we're looking at several different Homeobox genes, right? OTX2 being one, and then there are these other genes that are being expressed in the forebrain, the midbrain, and the hindbrain. And we'll talk about it some more in a moment here, but the hindbrain will later in development show these very obvious, nope, I don't have it there, show these very obvious segmentations. And we call those Rhombomeres. So there are several Rhombomeres that we just number from rostral to caudal. So when OTX is present, and GBX is present, the expression pattern looks like this. But if we knock out GBX, so see, no GBX there, notice how knocking out that one gene totally changed the pattern of gene expression of the others. So this is showing that they're interacting with each other, and that normally they're interacting with each other to make very specific segments of the nervous system. They're going to go on to take on a very different fate. And one of the things that stands out when you're looking at the gene expression of these genes is there's a big, sharply defined boundary between the midbrain and the hindbrain in gene expression, and as we're going to see in a moment, in terms of fate. So I didn't bother to have artists redo this, so it's ugly. But I'm just pointing out to you that the various Homeobox genes that are colored here, color-coded, are homologs of one another from fly and mouse. And you can see that the similarity is, immense, inescapable. Yes, the same gene family that is segmenting the Drosophila brain is also segmenting the mammalian brain. And don't bother memorizing those names down there. So in looking at the midbrain, it turns out that that we know something about the signaling that's involved, that something called fibroblast growth factor, FGF, warning, there are really like 8 different FGFs, and I don't ever want you to remember which number is which in this class. And so what they showed was that there was pretty good reason to think that it normally tells cells to start making a midbrain. So they soaked a plastic bead with a particular FGF and stuck it between the host midbrain and the telencephalon. And lo and behold, this animal on that side where the bead was made a second midbrain. And it's shown here, right? Here's the midbrain normally in embryos at this stage and chicks. And here's what happened with this chick, where there's two different midbrains, or you can look at it diagrammatically and you can see that one of the midbrains even started to make some cerebellar cells at a later stage of development. So it was inducing a new midbrain. And people went down this pathway because people found that they could take chick embryos and quail embryos, and they could take out a little bit of quail embryo nervous system and stick it into a chick embryo and induce a new set of midbrain. So just as there's the organizer that organizes the whole nervous system, or the optic cup induction, it looks like there are factors here that induce the cells around them to start making a midbrain. Good.
Did lice ever survive? Like, you kind of additionally spread?
This was all chick, and I don't know how far they get. I mean, probably. Well, let's see. We know in this case, they killed the chick at this stage, because that's how come we're seeing it. So my guess is many of these would be fatal. And when you're doing these kinds of experiments, the trick is to check in on various embryos at various times and make sure you get to them before they die. I don't really know the details of this particular experiment, but that's how that works in general. I have done chick embryo manipulation, and you definitely want to, keep an eye on at what point they're still developing, and you stop them at that stage of development and see what you can learn. Right?
Where is FGF secreted from, and then where did we find them to?
We're gonna run into FGF again later, but its expression pattern is complicated, which is why I didn't put a figure together of it. And also, it's not as easy to keep track of because it's secreted. Fibroblast growth factor, it has that name because the first people who studied it realized that it caused fibroblasts to divide a lot. So it's a great question, but I don't have a great answer. My fault. We can also look at the telencephalon and see various homeobox genes that are expressed in very specific patterns. So here's an example where they've taken two different homeobox genes, EMX2 and PAX6. I don't care what they stand for. And you can see that one's being expressed in the developing telencephalon rostrally, and the other's being expressed caudally. And in normal mice, here's the pattern of cortex in terms of the function they're going to take on. Motor cortex in the front, somatosensory cortex, auditory cortex, visual cortex in the very back, right? Just, pretty much as it is in people too. And if you knock out EMX2, so you've interrupted this gradient that's there, the different parts of the cortex take on change in terms of how much they take on various roles. So without EMX2, this would be an animal with loads of motor cortex and still somatosensory cortex, but hardly any visual or auditory cortex. And plus, they're not only smaller in size, notice they're reorganized in terms of where they occur in the nervous system. Or you do complementary knockout PAX6, and it's yet a different pattern. And now you have an animal with devoting a whole bunch of its cortex to the visual cortex, which, as you know in mice, isn't that important for getting around in the world. So my point is, these same homeobox genes that are regulating and specifying the fate of various parts of the fly nervous system are also doing that in the mammalian nervous system, even up to the cortex. Questions about that? Good.
So if you had the axes of PAX6 and you didn't have the motor, the somatosensory, and the auditory, would it just fill the entire forebrain neck?
I mean, so this is an animal without PAX6, right? So this is a knockout. Minus/minus means both genes are dysfunctional. And so no, it still has those regions. It's not totally taken over, but you can imagine that if you knew which players were involved, that you could end up with mutations that would indeed make a cortex that's all visual cortex. Or, tell me what the rules are and let me get in there and mess about with it, and I'll make an animal with all auditory cortex.
But is it like the organizers that are responsible for that? They can only go so far, spread so far, or?
Well, it's a complicated, it depends. It depends on whether the gene happens to be a transcription factor itself, as Homeobox genes are, or whether it's a gene that is involved in the cell-cell signaling between cells that directs their gene expression, or it's a product that is secreted and therefore, as you say, can go a certain distance but not too far. All of the above. All of that happens. And I'm not going to give you examples of all. Okay. I told you we were going to talk about rhombomeres. So here we are again, looking at the developing brain schematically. And the hindbrain consists of 8 different rhombomeres, and they are visible. You look at the embryos and you can see these swellings that look like discs lined up on a bracelet, right? Or that necklace candy where you have a bunch of candies on a string, right? You can physically see these divisions. And so anatomists, developmental biologists long ago called them rhombomeres. And they're numbered R1 through R8. And they are, of course, on the other side, they're the pharyngeal arches that we talked about before, and they happen to be numbered P1 through P4. So far, so good. During development, there are certain cells that are expressed in one rhombomere, but not another. And this happens to be a transcription factor called CROX. You don't need to remember that. But what you do need to know is that these cells are also expressing something called ephrins. So ephrins are membrane-bound proteins that the ephrin bound in the membrane of one cell can interact with a receptor on its neighboring cell. And we call the neighboring cell, what it's making, unimaginably enough, the ephrin receptor. We're going to run into ephrins several times. They're very important players in brain development. And here we're looking at the cells that are expressing CROX, and they're being expressed here in this sort of pattern early in development, but as development continues, suddenly they're being expressed in very tightly demarcated areas. And so 2 things are happening as development proceeds, which is some cells are changing which genes they're expressing. And in some cases, some of the cells that are expressing that are in the wrong place will actually migrate and be with their fellows. So both sorts of things are happening. Cells that are interacting with one another, either because they're signaling each other to direct gene expression and or because they're moving around. At this stage of development, everybody's capable of moving. And at this stage of development, these cells all want to be around the cells that are like themselves. And as that happens, then the line of demarcation comes into focus, right? And we'll run into more migration later. We'll see all of these examples again. And some Hox genes, the only Hox mutations I know of in humans that duplicate a body part are these cases where people end up with an additional finger, additional earlobe, and things, additional toes, things like that. So here's a Hox gene mutation that results in polydactyly, many fingers, many digits. And here's, and sometimes the excess digit is non-functional. It might just be another digit that's sort of vaguely formed on the other side of the pinky. And often surgeons, maybe even at birth, will simply cut that off because you don't need that. But in some mutations, they end up still being functional. And so here's a young man with 6 fingers, right, on his hand. And apparently he has use of all 6 And you're probably wondering, so is this a second index? Is this still the index finger? And I don't know the details of that, but it can happen. Okay. Again, Hox genes, what the weird stuff they were doing in flies is not irrelevant to what happens to people. Okay, I can tell you want another Kahoot. It's just obvious. So we've talked about how the nervous system in both flies and mammals is divvied up from anterior to posterior, right? That's what all those maternal polarity genes, et cetera, did, is they end up telling each cells where they are in the neuraxis, right? The neuraxis, right? Whether fly or mammal, that long string of cells that are nerves, spinal cord and brain for us. So it tells the cells whether that's towards the front of the neuraxis or towards the end of the neuraxis or something like that, right? And those cells are important for that. But that's not the only division of labor in the nervous system. There's also a big division of labor of dorsal and ventral, right? As you know, the dorsal spinal cord is important for sensory processing, and the ventral spinal cord is important for motor processing. And so now we're going to talk about how that happens. Oh, no, we're not. Yeah, we're going to do some more about posterior, more stuff about, yeah, that's true in mammals. So in addition to the Hox genes, there are also secreted factors that are telling cells where they are in the nervous system, where they are in the neuraxis in the anterior-posterior domain. And some of them are our old friends, the BMP antagonists, also known as organizers, which end up being more concentrated in the front end of the brain than in the rest. And so that's the BMP antagonist, whereas BMP itself is having more influence in the posterior end of the animal. And there are at least two other important factors that tell cells how posterior they are. FGF, they tend to be concentrated in the posterior end, as are the Wnt genes. And so these are all secreted factors that are getting made different places, different cells, that are passively diffusing around in the neuraxis and offering cells a cue as to where they are in the nervous system, and therefore what particular roles they should take on, which is to say what particular genes they should express, which is to say which way they should differentiate. And then there's another secreted factor that's different from the others, that's retinoic acid. Retinoic acid is not a protein. We'll talk about its structure in a moment, but it's not a protein, and so its expression in the caudal end of the animal is happening because the proteins that generate retinoic acid are more expressed in the posterior end. So here's an example of how Wnt, a secreted protein, how it interacts with cells. And I do want you to know that Wnt interacts with frizzled, but in terms of what happens after that, I don't need you to remember that pathway at all. I just want you to know that it ends with changes in gene expression, right? So it's going to change gene expression, and the further posterior the cell is, the more Wnt signaling it's going to get, and the more its gene expression is going to be pushed in the direction of, let's make something at the caudal end of the nervous system. Or for questions about that. The other caudal signal I talked to you about, mentioned, is retinoic acid. So here are some in situs of expression, not of retinoic acid, but of the enzyme that makes retinoic acid. Retinoic acid is part of a steroid receptor superfamily. So retinoic acid, it's not technically doesn't have technically the structure of a steroid hormone, but it's similar enough and it has a similar mechanism of action. And in fact, there's reason to think that retinoic acid signaling might have happened before any other steroids were around. So retinoic acid, here's the structure, and it is a very lipophilic, like steroid hormones, it's a very lipophilic molecule, which means it can readily pass through membranes. So it's being made by some cells that are expressing the genes needed to make retinoic acid in the caudal end of the animal. And then once it's made, it can diffuse. And there's not much of a circulatory system in this case. So mostly it's acting pretty locally, because that's how far it can diffuse. And retinoic acid, once it gets inside a cell, it alters gene expression by binding to a receptor. And that is a protein receptor. This is not to scale. Retinoic acid is tiny and the receptor is huge. And the retinoic acid receptor is a part of the steroid receptor superfamily. And we'll run into other steroid receptors when we talk about hormone signaling guiding neural development in chapter 7.5. So far, so good. So, like other steroids, the way it works is when it binds to the receptor inside the cell, that changes the conformation of the receptor, and now it starts binding to DNA and affects gene expression. And you might ask, which genes are being affected by the retinoic acid receptor complex? And the answer is lots of different genes. And for the gene expression to be affected, the retinoic acid receptor complex has to recognize a particular sequence of nucleotides in the DNA, a particular sequence of G's and C's and T's and A's. Got that? And just to make the terminology complicated, that particular sequence of nucleotides is known as the retinoic acid response element, RAR. It's madness. The chemical is RA, its receptor is RAR, and part of the DNA it binds is the RARE. Too bad. We're stuck with it. And then the genes that are binding, some may increase their expression, some may decrease, etc. And typically, there are lots of different genes being influenced. So here's an example where someone just put more retinoic acid inside the chick embryo. And remember, I told you retinoic acid is normally expressed more in the caudal end, or the rostral end. By raising the entire level of retinoic acid in the developing chick at this particular point, some of the cells in the back, in the middle, got the signal that they were supposed to make posterior stuff. So here's the distribution of cranial nerves in wild-type animals. In the animals exposed to RA, this rhombomere, number 3, starts making facial nerve-type connections, when it's normally supposed to be making trigeminal nerve connections. The details about here of the various Hox genes that are being regulated by retinoic acid, you don't need to remember that. I'm just telling you that if you add retinoic acid to any vertebrate embryo, you will end up posteriorizing the nervous system. Various parts of the nervous system will hear that signal. Oh, I must be further back in the embryo than I thought I was a moment ago, because now I'm getting more of the retinoic acid signaling, and therefore I'm going to make more posterior objects. How's that? Good. Justin, right?
Do they always have receptors for the
it's a great question. Not every cell has receptors for retinoic acid, right? But during development, it's almost universal, right? As development proceeds, the number of cells that are expressing the receptors goes down, And so if retinoic acid is around, it will get there, it'll get through the cell, but if there's no receptor for it to bind to, then it has no influence on development. And the retinoic acid will just drift right back out again.
It's like why cells can't like change their fate twice because something is receptor.
Well, once cells stop expressing retinoic acid receptor, now they've made themselves immune to it. They're not listening anymore. And indeed, early in development, almost everyone makes it, and as development proceeds, the number of cells that are continuing to make the receptor declines and becomes more specific. Different parts of the whole body, not just the nervous system, but we care about the nervous system. Well, it's an incredibly powerful signal, and that has problems. So retinoic acid is an excellent treatment for acne. So acne medication kind of underwent a revolution in the, 1980s or so, where suddenly, for the first time, they actually had an effective treatment for acne. This stuff, an ointment with this stuff, so you just rub it on. But because retinoic acid is itself very lipophilic, putting it anywhere on the body, the retinoic acid is going to find its way into the circulatory system, and it's going to be there. For most people, that's of no consequence. The amount that's going out, the amount that gets to the rest of the body is too trivial to make any difference. And indeed, for an adult, there are almost no structures that are listening to it, right? Because almost no cells are making the receptor. But if the person using the acne medicine happens to be pregnant, there will be plenty of retinoic acid to go in and alter the development of the embryo. And so it is a tremendous teratogen, which the roots of it is to make a monster. So embryos that have been exposed to retinoic acid have very severe birth defects. And it took a while before people realized that was the case. And so that's why, do I have a picture of it? That's why these days, the label says all over it, do not take if you're pregnant. And it should say, do not take if there's any chance you could be pregnant. Because, of course, the whole problem, don't get me started, is people are often pregnant when they don't know it. People being, women. Anyway, so how's that? So that's why that label's there. It is mimicking a very powerful developmental morphogen, something that is altering the structure of, in this case, the developing nervous system, developing body, but also especially the nervous system. How's that? So far, now, caught up. So now we've specified the neuraxis in terms of the anterior-posterior end, right? Every cell knows where it is in the anterior-posterior axis based on, for example, how much retinoic acid it's being exposed to, how much Wnt signaling it's getting, how much FGF signaling it's getting, how much bone morphogenetic protein antagonists it's getting, which are more concentrated in the rostral end, et cetera. And that can then tell each cell, give them some idea of what they should make next, which is to say, we'll direct them what to make next. But we haven't yet gotten to the dorsal-ventral segment, dorsal-ventral specification, which is also important for the spinal cord. So normally during development, as I've said before, there is that structure made of mesoderm that coalesces to make the notochords. And you've heard the notochord before because it's the presence of a notochord that is eventually going to give rise to the vertebrae. It'll be part of the vertebral column, and all the vertebrates have a notochord. That's what distinguishes them from other organisms, right? They all make this notochord, and made from mesoderm. And the notochord is releasing signaling factors. So in embryological experiments, people noticed that if they took a chick embryo and removed the notochord, that no floor plate was formed. So in a normal embryo, the notochord's here, and morphological changes happen in the developing neuraxis that look distinct from the rest, and makes it look like a foreplate. And that's going to be the ventral surface of the spinal cord. And because motor neurons are present in the ventral spinal cord, there's normally also a development of cells that become motor neurons. And motor neurons are easy to keep track of because they're among the earliest neurons that kick out of mitosis, and they right away get really big. So for several centuries now, we've been able to keep track of motor neurons because they're so large and such a distinctive shape. But if you remove the notochord, neither the foreplate forms nor do any motor neurons form. And at first, people were moving around notochords, and if they took a notochord from one chick embryo and put it next to the nervous system of another, they could induce a secondary foreplate and a secondary set of motor neurons. So they reasoned that the notochord must be secreting something that tells the overlying nervous system to make a foreplate and start making motor neurons. And when they did a screen of various things that notochords make, they found something called sonic hedgehog. Now, I'll take a little bit of time to talk about the terminology here. First, in flies, there were a whole bunch of genes that were found where mutations caused the flies to have funny hair patterns and made them look kind of like hedgehogs, right? And so, okay, whoever finds a gene and, all that stuff gets to name it first. And eventually people found other variants like that were clearly related to that gene, but had different phenotypes. And of course, this was when Sega was making big inroads with Sonic the Hedgehog. So eventually some wag named the gene he found Sonic Hedgehog, right? Just trying to be funny. Whimsical stuff is all over fly gene name. Okay. As we'll see, there are times when the whimsical origin of those names is going to cause trouble. Get there in a moment. So Sonic Hedgehog, and Sonic Hedgehog, they're secreted, and they bind to the external domain of sensitive cells. And in binding the extracellular domain, they alter gene expression. Now, I want you to know that Hedgehog binds to the patch-smoothened complex. And I want you to know that it alters gene expression, but I don't need you to remember all the details of how it alters gene expression. And it turns out that when the notochord induces a new foreplate and motor neurons, Sonic Hedgehog is a signal that's responsible for it. And the way they found that was that they, instead of putting another notochord there, they got COS cells that can be grown easily in a disc that don't normally make Sonic Hedgehog. If they transfected them with Sonic Hedgehog, so they were making it, lo and behold, the cells were just as good as inducing a new foreplate and motor neurons as the notochord was. And so you end up with a situation where there's, in addition to the rostral-caudal gradient of signals, there's also a dorsal-ventral gradient of signals. And one of the powerful ventral signals is coming from the notochord, and it's Sonic Hedgehog. So it induces the part of the nervous system that's closest to it to make a foreplate. And now the cells in the foreplate, they start making their own Sonic Hedgehog. And they have influences on the cells next to them. And eventually, some cells, if they get just the right dose of Sonic Hedgehog, a small number of them will start differentiating into motor neurons. There's also a dorsal signal. So remember the ectoderm, right? Where everyone's secreting bone morphogenic protein and telling each other, we're going to stay ectoderm, we're going to stay epidermis, we're going to make skin someday. They're continuing to secrete bone morphogenic protein. And so they induce the part of the nervous system closest to them to make a roof plate. And the roof plate then starts making another set of proteins, so that you end up with this gradient that by the time you look at all these factors together, a cell will know where it is in the dorsal ventral plane based on what combination of these factors it's getting exposed to. And that's going to change the fate that they take on. So when we get back Thursday, we will, when we get back next time, we'll talk some more about these gradients and what they do, and so a little bit more about what Sonic Hedgehog does, and then we'll move on to chapter 3. All right. Thanks. Questions? Oh, darn, I got my signals mixed up. Well, I'm letting you out 5 minutes early. I didn't mean to. I'm not normally that nice. Okay.
I don't know. I thought they are so much.